He's one of the PIs of the Center for Biodefense and Immune Modeling.
Okay. Jing is our program manager, actually. If you have any questions about this program and also Symposium, you can ask her. And as Jing mentioned, myself and Martin, who is sitting back, we are the co-PIs for the Rochester, Center of Biodefense Immune Modeling. And for this summer school and also the Symposium, follow this, start from Thursday. It's required a program by our, NIH sponsor. And today, first I will give you a brief introduction and overview about summer school, and also I will briefly mention the Symposium also. And actually, before I do that, I will introduce some basic concept about computational immunology, because this is the focus of our center and also our focus of the summer school and the Symposium. And then I will talk about this multidisciplinary interplay for this new area. Actually, this computational immunology is kind of a new emerging fields, so that's why I will introduce these concepts first. Then we'll discuss something like why immunologists need to learn this modeling. I think most of you are kind of like experimental immunology background, and so we, when we try to screen the candidate for this summer school, for this summer school in particular, we are targeting at experimental immunologists, or at least you have an immunology or virology background, not mathematical, not computational background. So then I will talk about why you really need this. And then I will just briefly talk about the summer school overview and what we will do next couple of days, and also talk about the Symposium. And now let's discuss, what this, kind of fancy terminology means. Because right now, when we talk about computational biology or bioinformatics or system biology or even computational immunology, whatever this terminology jump to, in front of you, it's kind of vague. There's no clear definition what this means. What this, this terminology or these fields requires, what kind of background requires. Seems like many different kind of people are doing this kind of thing. So I will discuss this briefly, and then I will talk about how we use this terminology or use these concepts to do our research. And the first thing is this very very broad definition about this so-called computational biology. If you go to the wiki to look at the internet, you can Google it, you can find the definition. Basically, you find like this is like kind of interdisciplinary field that applies the modeling, analysis, and computing techniques from all different kind of areas. I expand that definition a little bit. Probably in the Google definition, probably we'll find like they may just use computer or informatics or whatever, statistics techniques. I just put all this like computational science or mathematics, statistics, informatics, physics, even engineering, to address biological problems. So basically what this means is any of this kind of like computational, quantitative, or modeling or data-oriented techniques can be used to address biological questions. These people usually call themselves doing the computational biology. That's why people tell you I'm doing computational biology. Some of them are computer science background, some of them are like mathematicians or statisticians, and physics people, even engineering people. And so this is a very broad, definition and quite a diversified area. So this is the general computational biology. Basically, you use any computational quantitative science, techniques to deal with biological questions. You can call yourself computational biology. That's what we are doing too. And another term is bioinformatics. What's the difference? If you look at the bioinformatics definition, also is a kind of like interdisciplinary field that also apply this computer science, statistics, or information technologies to the field of biology and biomedicine. And particularly this one a little bit emphasize, particularly to the analysis and annotation of genomics, genetics, and other kind of high throughput omics data. That's why this field is compared to the computational biology. This is like more like dealing with the data from genomics level or genetic level, and also the high throughput. They call for those people using used computer science or statistics methods. Usually for this one, information technology usually also mean that, from my understanding, is also computer technologies. They call it information technology. Like you develop computational tools, softwares, or even kind of database to deal with the high throughput data. They also call themselves the bioinformatician or bioinformatics. So this is the bioinformatics definition. And basically, when I talk about bioinformatics, this includes they call development application of all these different kind of things. If you look at the journals, journals or associations or the people who are doing this kind of bioinformatics, they are trying to develop like computing algorithms and softwares and the databases, as I mentioned, maybe even some artificial intelligence techniques or web technologies, computer simulations and data and text mining, and also machine learning, so all this kind of computing related technologies. So this is the one area or sub area of this bioinformatics. Basically this is developing all these things from a computational perspective. Second thing is definitely is a statistical sub area. I'm a statistician actually by training, and also this includes like pattern recognition and some other like statistical data mining. All these kind of methods. So this is also another sub area of this bioinformatics. And definitely another thing is like use mathematical other computational modeling. This modeling means you probably do more kind of complicated modeling, like network models. Like here we do a lot of different equation models, to deal with deal with the biological information and data at all these genetic, molecular or protein or cellular level. So this is more like still you can see this bioinformatics is also cover very broad area. You can see this. And another terminology is so-called system biology. This started probably like late 1990s. And so when people talk about system biology, I'm not sure how many of you know about system theory. They call system theory. Basically, intuitively, system biology means you have to consider a biological process or a biological system as a system, really as a system. What that means, system as a system. Basically, you can see also this is a kind of like interdisciplinary field that focuses on the systematic study of, one thing is this try to deal with complex interactions between the components, elements of biological system. So that means this component means like a molecular level, genetic level, or cellular level, whatever, different levels. Each of this level, or each of the gene, or each of the protein, or each of the cell can be considered as a component. The question, so this system biology is trying to study their complex relationship or interactions instead of just focus on one gene or one protein, one cell to do just one thing, but we try to look at the interactions. And also try to look at how these interactions give rise to the function or behavior of the system. So that's another thing. Besides studying those interactions, and also these interactions instead of individual components may have an impact on the behavior and the function of the system. So this system biology try to look at the interaction and the behavior and the function. And also a very important concept here is to use so-called integration rather than reduction approach. So this is the key point, which is different from a traditional experimental perspective. Traditionally, most of you are immunology background, and when you do immunology experiments, what you are trying, one lab usually try to do is, if you are focused on a T cell, for example, experiments, you try to design experiments just to look at the T cells. You ignore many other things. Or if you are interested in one gene, you try to knock out that gene. Use mice or whatever. Just focus on knock out this gene or wild type, compare them. So you try to make a very complex system into very simple binary comparisons. That's a traditional way of thinking to design experiments. When you try to do the experiment, you try to narrow down to think about, if your lab can, just deal with this T cell or B cell, whatever, even a subset of that. You just try to narrow down everything, look at it as one thing. And this is a traditional so-called reduction approach, try to simplify things to work on it. And this system biology approach, this new concept is try to use this integration concept. Integration means now try to put all the pieces together to do it. Instead of just look at one gene, one protein, or one type of cell, and you should look at the whole system that related to this biological process. Like we are working on the flu infection. So when you try to study the flu infection, if you do experiments for mice, like we do, we inject flu virus to mice, or just use whatever the different way to give them flu virus to mice. Then we are trying to look at how the immune system responds to the flu virus infection. And when we do this, the integration way to do this, not only we are looking at different type of cells, like B cells, CD4 T cells, CD8 T cells, antibody, all these kind of key elements of immune response. We have to look at all these things simultaneously instead of to reduce it into just one lab to do it. In this way, usually maybe one lab only has the expertise to do one type of cells. You are expert on CD4 T cells. Probably, you don't do too much on CD8 cells, even dendritic cells or B cells. And in this, when you do integration, one lab probably cannot do it. So you need multiple labs to work together to look at the whole system. What's the key element there that simultaneously do it, get the whole system, to look at how this whole system works, not just the one piece of the element works. So this is the concept of the system biology approach. When I talk about this system biology approach, actually, also this in particular also not only from experimental perspective, you need to use this concept of system biology, but also this require a lot of quantitative science technologies. And here, like I list here, so this includes like development and also application of this, like control theory or system theory. Basically the system theory comes from engineering concept. If you look at circuits, in the electrical engineering or the control theory, people particularly study the property of some kind of engineering system. So here you just use that, engineering concept, system theory concept into biology. So that's why you do need to, use some kind of this control theory or system theory concept to this biological field. And also this quantitative modeling of biological process, definitely we need mathematics, engineering, physics to link all these pieces together. When you do the traditional reductionist experiments, usually you just compare two things, like wild type versus mutant, or knockout versus the without knockout, or treatment versus control. That's the traditional way you are thinking to compare these things. When you do this integration way, system biology approach, because we are looking at all these components together simultaneously. So the question is, how can you link all those different kind of data, different kind of elements together? So that's why you really need the kind of complex quantitative approach to link the data together, link all these different, the function effect of these different things together. So that's why this is critical. And also, when you finish your experiments from the system biology experiments way, and you need to integrate all those data together. This you need kind of advanced statistical methods to deal with the data. So so this is the basic concept of the system biology. So. And also, when you look at again, this is just repeat. When you look at the system biology study, the key feature and also from the experimental design perspective, all these things are very important. I just mentioned that one thing is definitely when we try to design a system biology experiments, we are trying to study the interactions. So that we need to capture the dynamics feature of the system, and this need what this means is when you try to capture the dynamics, means, for example, when you try to look at flu infection, you inject the flu virus to the mice, then the immune system starts to work, to respond. And this responding, this response actually, the response is a dynamic process. It's not just a one-time point. It's just basically, from the beginning, start to respond, then there's a whole process, go up and down, whatever. There's a whole dynamic process. So when you look at these interactions, then we need to capture all this dynamic process. Then in this way, we do need, design experiment to collect the time course data, not only one-time point. So that's the traditional way, just look at one-time points, or even probably 2 or 3 time points, but not the whole dynamic process. So this is one of the key features for systems biology, study. And this idea I already talked about, integration rather than reduction. Integration again means we have to measure all the elements of the system simultaneously, instead of like one lab do one thing, another lab do another thing. One lab do the CD4 T cell, another lab measure the CD8 T cell, but these two things are different, system. If you measure one, measure CD4 T cell from one mouse, another lab to do another one from another mouse, this not connect each other. So we have to measure this, simultaneously. This is another thing. And this is also I mentioned that definitely this needs more advanced data analysis methods to integrate with this kind of mechanism-based models. When you try to use a mathematical model to link all these components together, and then the existing statistical methods may not work to deal with the data. So that's why in this summer school we also have one-day lecture about statistics. Probably we don't have enough time to teach you too much of those advanced statistical methods, but at least give you some feel or give you some basic concept how to use statistical methods to deal with those data. And definitely for this kind of system biology study, the cost is high. When you are trying to look at many things simultaneously, and in terms of the labor, coordination, how to do this, this is the more expensive than the traditional reductionist experiments. So this is the thing. And when we talk about here, we are focusing on immunology. So if you just borrow that concept of computational biology into computational immunology, or whatever you call from bioinformatics into so-called, right now people call this immunoinformatics, whatever. You just to change the word from biology to immunology. And I just try to summarize this to use based on our experiment. Based on our experience in the past 6 or 7 years working on this, I give it a kind of definition for computational immunology or immunoinformatics. We just try to use mathematical computational models or representations, and derived from immunological or biological concepts and mechanisms based on experimental data to query or address immunology problems by employing computational methods, bioinformatics, or system biology approaches. Which I try to use all the methods, the concept that I just mentioned, because all those concepts, I talk about computational biology, bioinformatics, and system biology. You can see all those concepts have a lot of overlap. They definitely have different perspectives, but basically, you need to use a lot of quantitative, computational, or mathematical, concepts into biological experiments or biological research. So I just borrow that into immunology. So here is basically we are trying to use these mathematical computational models, but all these models are derived from the basic immunology or biological concepts and the mechanism, and also based on data. So it's not just the theoretical mathematics. So this is the thing we are trying to talk about. And also, here, in order to do those things, we need to develop and also apply the methods. Again, I just summarized all these quantitative or computational technologies from computer science, even mathematics, physics, engineering. The computer science basically definitely you have to deal with computational issues, simulations. And this mathematical physics, engineering, basically we have to represent the experimental, all immunology experiments into a mathematical formula. So that's why we need, sometimes we, mathematics and maybe physics and engineering also, use some of the physics engineering concepts to construct the mathematical models. Statistics definitely to try to link this mathematics to your experimental data. When you deal with the data and you also have a model, you link these two things, then you need the statistics. Informatics is basically, how to handle the information you collect, the information, the data you collect from, this kind of, computational immunology experiments. As I mentioned earlier, when you do this, probably you are looking at many elements at different levels, at the genetic level, molecular level, protein level, cellular level. So in this way, you may apply many new biotechnologies, like those omics, high throughput technologies. You collect a lot of data and a lot of information, not only the purely number of data, but a lot of information, how you collect those data, like those protocols, and how you produce those data, and each of those technologies require a lot of work. Once you get the raw data, you need a lot of work to process those things. You finally get one number, so that's also a lot of information, how you do that. If you use different way to do it, you may get different numbers, So a lot of information you need to manage. You need to put those information somewhere so that you can easy to access, easy to share, easy to retrieve, to do further analysis. So that's why you also need informatics things, So we need to use all different this kind of computational, mathematical, statistical technologies to deal with immunology problems. So this is the concept. It looks like all these things is quite complex, which is true. Here I just put a figure, what we are trying to do here, locally. We are, this is when you do, computational immunology study, you do need a multidisciplinary team to collaborate, with each other. Here in our Rochester Center, we have different kind of people. Definitely, we have our immunologists. We have 7 or 8 labs who work with us. Those different labs have different expertise. Some lab are expert in CD4 T cells, as I mentioned. Some lab is interested in B cells, dendritic cells, or different kind of immunology and virology also. And so we have 7 or 8 labs collaborating with us. And definitely there are a lot of there are postdocs, technicians who do a lot of experiments, collect a lot of data. And so this is the these 2 red boxes basically experimental, and I emphasize more like this quantitative computational components here, and I call this is the quantitative integration. We need we need people like who are bioinformaticians and statisticians to do statistical analysis. We have mathematical modelers, then we have computational people, and then we have data management, database data management people, and then we have software and tool development people. And then here is the IT support, So all these people, I put the arrow, probably cannot see the arrow, which direction it goes. Basically, this arrow goes to the up, So basically, how we support from here, this quantitative integration support all the immunologists, definitely our database people try to manage the data from, the data collected from the experimentalist, and definitely all these bioinformatics, statistics, mathematics, and computing people, we have to integrate all this together to support our immunology research, to address immunology questions. And definitely all this computing support for all these things. So this, this is like complex teamwork or complex network to deal with a complex biological network. So we have a human network, it's quite complex, because we are dealing with a very complex, biological system. So that's why we really need these multidisciplinary interactions. I know when people talk about something is very complex, you always say this is not a rocket science. I can tell you that the rocket science is not as complicated as biological science. I think the biological process is much more complicated than rocket science. So for rocket science, if you know that for rocket science, when people try to design one rocket, usually it takes probably 200,000 to 300,000 engineers to work together for 10 to 15 years to design one rocket. I'm not sure how long it takes for experimentalists or whatever to study one biological system. That's for rocket system. You can see the investment and the labor and intelligence from 200,000 to 300,000 engineers. Those engineers are different kind of engineers. Not only pure they have mechanical engineering, electrical engineering, computer engineering. Different material engineering, whatever, chemical engineering, all those people have to work together to design one system, one rocket system, and it takes 10 to 15 years. I don't think any immunology or biology project like that, you organize so many people work together to study one system. It's not. But however, any of the biological system, even small as a cell or single cell, is much more complicated than the rocket science. Just people don't realize that. When people try to design rocket or design airplane, they have a very well organized team. It's more complicated than this. Definitely want to do that. But in biological research, usually people don't recognize they also need this kind of effort in order to understand or deeply understand one biological system. And so that's right now when we talk about system biology, people start to learn, start to recognize that, try to put more money into one system, one project, so that we can understand one biological system in a kind of like organized way, not like piece by piece. So that's the thing. Now I just try to talk about why. I talk about already talked about this question actually. Why as an immunologist you need to learn modeling. Why you need to learn mathematical modeling or computational modeling? Why you need to learn all those quantitative technologies? It's sometimes those things probably we'll see is kind of boring. When I talk to experimentalists, when I told them I'm a statistician, they said it's a boring job. So why you need to learn all this? Sorry. Here I list, some of these reasons. It's not an exhaustive list, but from my understanding, why we need mathematical modeling or computational modeling for immunology research. Number one, definitely, this modeling just help you to quantify the immune processes. When you work on the immunology research, and when we have, there are several recent program announcement from NIH. They try to put more money to bridge mathematics and biology, or mathematics and immunology, whatever. They try to do this. When they have that program, they particularly mention that for any biological fields, if you cannot quantify, then they think that field is not mature. So NIH try to put more money to speed up the maturity of that biological field. So that's why if you say, my field is so so many uncertainties, I cannot quantify. Everything is qualitative. It's not quantitative. That means that field is not mature yet. In order to make that field mature, you really need to quantify that. You can measure things. You can really, to quantify the relationship and quantify all the things. So this is the first thing. You need to quantify the immune response. Number two, definitely, as I mentioned earlier, when we talk about system biology, maybe I can talk about here is system immunology. Is we try to model the immune system as a real system, in a systematic way. A systematic way means you have to treat this immune system as really a system, not just piece by piece, not just one lab look at one piece of the immune system, another lab look at another piece. You never put them together. Here you need this quantitative modeling approach to put all these pieces together. So this is another thing. Number three, definitely when you use some kind of modeling or statistical approach to deal with the immunology experiments, you can design more efficient experiments. So if you think about how to put things together, and then you can, before you do your experiment, if you think about your how to quantify things, how to do the data analysis, then at the beginning you can really think about how to design experiments more efficient in order to address your scientific questions. So this is the thing, usually statistician can help you to make your design, like kind of optimal or more efficient. And also we try to use quantitative or computational modeling approach to extract more information from experimental data. When you collect a lot of data, you don't know how to link them. Usually people just simply get the simple conclusions. Actually, when you use more modeling approach or computational or statistical approach, probably can get more information. Originally, say I collect a lot of data here, I publish one paper. Probably if you using this modeling approach, you can get much more papers, many more papers. So this is the thing. Actually, when I worked, before I came to Rochester, I worked for AIDS Clinical Trials Network, in Boston, and I worked on one of the clinical trials that have maybe 40 or 50 patients. And originally that study, that PI tried to collect a lot of immunology data. Even he didn't know why. He just, I don't have an idea why I collect a lot of. He measured everything originally, initially every week, even a couple of days. Then later he measured, monthly or every 3 months. He collect like whatever, the CD4, CD8, memory cells, the naive cells, all this, use flow cytometry to get everything. And besides, he measured like the thymus, even the weight of the thymus, whatever he can measure, he just use it. He doesn't know how to use the data. He just collect whatever he can. That's it. And when I find that study, I talked to the PI. Do you know how to deal with? He said I just try to come, to do a distributive statistics and publish one paper. And they did. They published one paper. Then I started to talk to the PI. Say I can do a lot of modeling. Finally, I have that study. That team published maybe 30 or more papers. So you can he already designed that. Just one paper, but then his goal is, I want to publish more number of papers than number of patients in that study. That's his goal. So that's the thing. If you got a lot of data, if you use more advanced modeling or statistical approaches, probably you can really get more information than you can think about. So this is the thing, and also definitely. And also I mentioned, that in order to integrate and also interpret complex experimental data, you need this quantitative modeling approach. And also for simulation and prediction purpose, if you want to predict or simulate what if, you look at the what if questions. Some of those questions you cannot do experiments. You want to do predictions. Cannot do all the experiments. So you can do something, but for some other things you need to do prediction and simulations. So without a quantitative or computational or mathematical model, you cannot do this, right? And also we try to use this modeling approach to identify therapeutic targets and also try to design new drugs and vaccines. This modeling approach can help you to identify those targets. So this is a list, for the reason why, we need modeling for immunology research. And also, why you need modeling? Here is another thing. So right now, I know some of the postdocs, students are not faculty yet. You will become faculty soon, and so the grant pressure is a problem when you go anywhere in the medical school or in the basic biological department. And these 5 criteria for AH grant review you are very familiar with. They talk about when people review your grants, they look at your number 1 significance, innovation, approach, investigator, and the environment. And actually, when you also submit a paper, many of your papers will get rejected. Why? Also, because the paper also they look at this, whether your paper have a significant, you are try to address a significant scientific question, or whether you have some kind of innovative ideas and methods, and also whether your approach are appropriate. It's the same thing. You review papers or review a grant. These 2 things for the grant review, investigator and environment, you cannot change it. You investigate yourself, whatever background you have. You cannot do anything. Environment, you cannot do anything. If you are working in an environment or in a university, unless you change your job, otherwise, these two things is difficult to do anything. However, for these first three things, you can make an effort to do it. And this is what, I'm thinking the modeling, computational modeling, or do more statistics, advanced statistics, mathematical computational modeling, probably can help with these three things when you try to apply for a grant, or you try to, publish a paper. And in what sense, so one thing is like, we can, from modeling perspective, we can help with, one thing is, when you try to propose aims or try to define your scientific questions, okay, and if you can think about from modeling perspective, computational perspective, if you make your data analysis part novel, usually people think your aim are also novel. When you think about the aim, if you are just limited to say your quantitative knowledge or your analysis knowledge, just compare two groups, wild type versus the control, the wild type versus the mutation, or treatment versus control. If you just do this very simple design, so you compare two groups. One group is whatever, knockout, another one is wild type, and you just compare. This thing from experimental design or from the it's hard for you to think about more advanced, more novel aims or more complex scientific questions. So when you talk about complex scientific questions, you really need a complex modeling or analysis approach. If you know those approaches, this will help you to think about more complex or more novel scientific questions. This is the one thing. And second thing is, as I mentioned earlier, probably the modeling approach or more advanced statistical approach, can motivate you or can help you to design novel and efficient experiments to generate unique data. So the unique data means if you not just compare two groups, you design the two arms and generate simple 3 mice for each arm and then compare them using t-test. And that's too simple. And if you can design really some kind of novel, fancy stuff, it doesn't matter you are doing grant application or paper. These people always think from experimental perspective it's novel. Okay. So right now, I work with our immunologists here, in Rochester, and when I first, came to Rochester, that's 8 or 9 years ago, like 2003. And when I talked to immunologists in the department, we have like, I'm not how many, 40 or 50 immunologists in the faculty, in the department of immunology, microbiology. I asked them, who do you work with in the biostatistics and computational biology department? They say, no, nobody. I don't need, they said, I don't need them. I can do t-test, I can do it. No, I click something and do it. And nobody work with, statisticians or computational biology. And however, my background is more like immunology, infectious disease, modeling and statistics. Then I started to work with them. After, this 7, 8 years, after this, in the past several years. And now many of them come to me. So if they write a grant proposal, they ask me, oh, could you add something like differential equation or some fancy thing? Make my proposal really, attractive and novel. So that's we gradually change their view on this modeling. That's one of the purpose of this summer school, and also this is required by NIH. Our modeling project. They actually NIH push us. So for this education program, you have to promote or educate experimental immunologists or virologists to recognize the importance of modeling and computational modeling and mathematics or statistics in their immunology research. So we have to promote. That's what we did. Promote. Now many of them, try to see in their grant proposal, try to see can you do some fancy modeling stuff. That they think that really helped them to get a better score. For the grant. So which is good. And also another thing, last thing, Stephanie, I just mentioned also, if we can use novel analysis methods on the modeling approaches, we really can extract more information and also even unique information from complex experimental data. That's all I just mentioned, talk about this. Okay, and then I just come to the summary and for this. And basically, I think the computational modeling, approach is important and also very useful for immunology research from whatever you just, do your research, publish, or from the grant application perspective. I think this is also always important and useful. But this thing, when you do the computational modeling, we really need multidisciplinary collaborations. Basically multidisciplinary, not only between experimentalists and the quantitative scientists, but also among the quantitative scientists. There are a lot of different things we need in computer science, information technology, statistics, mathematics, engineering, physics, bioinformatics, things like all these kind of different kind of people. Actually, even for all these people, they're more quantitative and computational, but still there's a lot of things we need to integrate. Even the people from computer science and statistics, they are using different languages. We want them to try to integrate, to communicate. Even that one is complicated. No matter you want to talk about all this integrated with immunology biology, definitely there's another step to go. So the question is how to do the collaboration, integration among all these different kind of disciplines. That's quite challenging. And the challenge is, again, so among computational quantitative scientists, I mentioned, this is the challenge, also already challenge, very challenge. And between computational quantitative scientists and immunologists and biologists, another step. And also lack of common language and culture. When you talk about computer science, statistics, mathematics, all these people have different backgrounds. Like mathematicians, they say, if I want to publish a paper, I like single author papers. I don't like you have like a biology, you have 20 people in a paper. And then in mathematical department, they think, if you have 20 people, your contribution is 20, just one out of 20, so they don't think that's your contribution. So, this is a different culture, right? And so how to deal with that? That's the problem. And definitely lack of integration leaders and experts. Each of these areas, you can find very good experts, computer science, mathematics, statistics, definitely from an experimental perspective. So the question is, there's no leaders or the experts really can put all these things together, because this involves 5, 6, 7, 8 disciplines. You need somebody who can understand. You don't need to understand each piece deeply, but at least somebody can put all these things together. So this is, I just talked about the rocket science, field. They have so-called general engineers, whoever, chief engineer, whatever. Somebody have to put all those 200,000 engineers' idea or whatever the technology into one system. Otherwise, rocket never fly, right? And so there's questions in the immunology or biology experiment, field. We don't have training in that to doing that. For engineering, they do have training for that, but this is not. So that's why we have this summer school, training purpose. We cannot deal with all these issues, from this just less than 1 week, several days, summer school. But we are trying to make our effort to try to bridge between computational quantitative scientists and immunology biologists. So here, for this summer school, as I mentioned earlier, we are trying to more focus to teach or promote quantitative computational concepts to experimentalists or immunologists, like you guys. And but some others, hopefully in the future, we can do the opposite way. Teach those mathematicians or computer scientists more biology. That's both ways. We need both ways. And so basically, the goal for this summer school, these several days, we will give you a light introduction about the computational modeling concept and analysis methods. We cannot, we don't have, too much time, to do a, deep teaching about all these concepts, but at least we give you some, introduction. And also we try to, hopefully you can learn the importance and the usefulness of modeling approach in immunology research, and also try to learn how to collaborate with computational quantitative scientists to use the modeling approach to deal with complex immunology problems. So this is our goal. I don't think we can do very deep, the mathematics, statistics, but at least you can learn some basic concepts, some basic things, how to collaborate with modelers and statisticians to deal with your problem. So this is the goal, and basically, this is the 5 days, the summer school. And then today, day one is today. Basically, we have lectures to give you some basic concept introduction, about modeling and analysis. And after me, Martin will talk about. Martin is a kind of immunologist, but he knows a lot about modeling. He can do different equations by himself, but but he will talk about from a immunology perspective, why modeling is really useful. I just talk about from statistical mathematical background. And then after Martin will introduce your computational concepts and the mathematical concepts. And in the afternoon, late afternoon, I will come back to talk about basic statistical concepts. So today, basically, like kind of an introduction about all these, computational mathematical statistical concepts. Then tomorrow on day two, we will focus on the differential equation models. Differential equation model is I think it's the simplest mathematical model to deal with the biology. So that's what also our Rochester Center's focus. We develop a lot of different equation models to describe biological process. So we will, the whole day we will talk about the, different equation models. And then day three, again, Tuesday when we come back, we will talk about more details about the statistical methods. Because when you try to bridge the mathematics and the immunology, what that means is you want to try to bridge mathematical model and your experimental data. That's the statistics. You always need statistics to deal with your data. So that's why we have a whole day to talk about statistics. And then the day four, we have a whole day talk about agent-based model. This model, probably some of you know that. This just use a computer simulation model, try to simulate the biological process. So, we will have two speakers on this. And the last day, actually, on the Thursday morning, we have half day talk about the flow cytometry data processing and the gating, because this is the critical tool. Flow cytometry. Flow cytometry is a very important tool for immunologists to use, but immunologists are struggling to how to deal with those data, like in particular for gating. So we'll whole morning we'll talk about using the software, computer software, to deal with the data processing and the gating. So that's the summer school. And the speakers, basically we have Martin I just mentioned, who is a co-PI there, and he will talk about after me. And Jackie Miao, I'm not sure he's here. He's not here yet. And he will talk about most of the today and tomorrow. He talk about the computational mathematical concepts, and tomorrow he will talk about different equation models. And then Hongmin Yang and Hua Liang and Xin Chu actually replaced by Rui Hu. And so plus myself, four of us, and also Jackie will talk about in the last lecture. This is the more statistics, whole day on statistics Tuesday. Talk about different kind of statistical methods. And Catherine and Tom Kepler, Catherine from Western University, Toronto, and also Tom Kepler, I think, from Duke, but now I think in Boston University. And the two of them will talk about agent-based models, computer simulation models for immunology. Okay, and Tim Mossman and Richard Sherman, Thursday they will talk about flow cytometry gating. So this is the two, and this is myself, and talk. And after the summer school, and that's on Thursday afternoon, and I think start from 1 o'clock, then we'll have a symposium. Actually, I think the symposium is kind of like advanced, introduction about the immunology, modeling. And so we invite like leading scientists, investigators from the world to present, their research in such that they can show some good examples how to use the modeling approach to do research, to do immunology research. So this, this is like a whole Thursday afternoon and a whole Friday, and we have 3 sessions for this. Thursday afternoon we will have a bioinformatics session, which is, this session will talk about using bioinformatics modeling approach for immunology research. And Friday morning we will have a session about bridging immunology and mathematics. How do you use mathematical models to represent your immunology system. And on Friday afternoon, we'll talk about bridging the data and the models. That's the statistics. So this is 3 sessions for this symposium. If you can stay, that would be good. Again, I will just acknowledge, this is our modeling grant. This is for Rochester, Center for Biodefense Immune Modeling Grant. This is the, most of the money we come from, plus some money from our deans, because right now the NIH grant cannot buy food for us, so all your lunch provided by our dean. And also, besides Rochester, we have other 3 centers. Center for Computer Immunology at Duke. I think right now they moved to Boston, and another Center for Modeling Immunity to enteric pathogens at the Virginia Polytech, and also another program in Mount Sinai. They call it Program for Research on Immune Modeling and Experimentation. So this all, this is the 4 centers for this biodefense immune modeling. We work together to host this summer school and the symposium every year. So next year, I think next year will be in Mount Sinai, right? What? Duke. Okay, that will be Duke. But I'm not sure they will be in Duke or Boston, because the PI moved to Boston. So we'll do this every year, and all these 4 centers take turn to host this. And this is for this year. This is the organizing committee. Myself, and Martin, with the co-chair, and Alan Person from Los Alamos, and Tim Mossman, Hua Liang, Hong Yimiao, and Jing over there. So all of us actually work together to put all these programs together. And also we appreciate the NIH project officer, Tim Gang Lewis, and Alison Ducott, and Ashley Xia, and I'm not sure Ashley is here, right? Oh, he's over there, cannot see. And Ashley is here, but Tim and Alison cannot make it because NIH cut their travel budget. They're supposed to come together, they cannot travel. So but we really appreciate them. They give us the money to do this. Otherwise, we cannot do this, right? So that's all I have.